విధానంలో డాక్టర్ బిఆర్ అంబేద్కర్ ఓపెన్ యూనివర్సిటీ దేశంలోనే మొట్టమొదటి సార్వత్రిక విశ్వవిద్యాలయం. దాదాపు 2.5 దశాబ్దాలుగా దూర విద్యా విధానం ద్వారా కంప్యూటర్ కోర్సును అనుసంధానం చేస్తూ బీఏ, బీకామ్, బీఎస్సీ డిగ్రీ కోర్సులను అందిస్తోంది. పోస్ట్ గ్రాడ్యుయేషన్ స్థాయి, డిగ్రీ, డిప్లొమా స్థాయి కోర్సులను ఔత్సాహికులైన విద్యార్థులకు అందిస్తోంది. డెవలప్మెంట్ స్టడీస్లో సెంటర్ ఫర్ ఎకనామిక్స్ అండ్ సోషల్ స్టడీస్ ద్వారా ఎంఫిల్, పీహెచ్డీతో పాటు విశ్వవిద్యాలయమే స్వయంగా దూర విద్యా విధాన అంశంపైన పీహెచ్డీ వంటి పరిశోధన కోర్సులు ప్రవేశపెట్టింది. రాష్ట్ర వ్యాప్తంగా సుమారు 4.5 లక్షల మంది విద్యార్థులు ఈ విశ్వవిద్యాలయం నుంచి ఉన్నత విద్యావకాశాలు పొందుతున్నారు. డాక్టర్ బిఆర్ అంబేద్కర్ సార్వత్రిక విశ్వవిద్యాలయం స్థానికంగా స్వయం బోధనా పద్ధతిలో ముద్రణ రూపంలో రూపొందించిన కోర్స్ మెటీరియల్ అందిస్తోంది. కోర్స్ మెటీరియల్ అధ్యయనానికి సహకరించే విధంగా రేడియో, టెలివిజన్ పాఠ్యాంశాల ఆధారంగా కార్యక్రమాలు ప్రసారం చేస్తోంది. అధ్యయన కేంద్రాల ద్వారా ఏర్పాటు చేసే సలహా సంసర్గ తరగతులు, సైన్స్ ప్రాక్టికల్ సౌకర్యాలు విద్యార్థులకు స్వయం అధ్యయనానికి ఒక చక్కటి అవకాశం. ఇవి విద్యార్థి సమీపంలో ఉండే అధ్యయన కేంద్రం నుంచే పొందొచ్చు.
Hello viewers, welcome to today's teleconference. First of all, let me introduce myself to you. I am Dr. Girija Nethi, Professor of Zoology from Dr. B.R. Ambedkar Open University. Today we have invited 2 experts to participate in this teleconference. Let me introduce them to you first. She is Dr. M. Rajashri.
Thank you, ma'am.
She is head of the Department of Zoology from Vivek Vardhani College, Jambag, Hyderabad. And she also has been the counselor in Dr. B.R. Ambedkar Open University. Thanks Dr. Rajashri for coming for this teleconference.
Thank you.
And another expert we have is Dr. P. Ramadevi. She is a scientist. She is from Bangalore, working in ITC and Research and Development Center from Bangalore. Welcome to you too.
Thank you.
Okay. Today's teleconference is actually for 1 hour duration, and for the 40 minutes, our experts will be talking about the subject. Afterwards, all you students are given 20 minutes time to interact with all of us, and please note down the numbers first. Take the numbers 040 2703 1431, 040 2703 1432, 040 2703 1433. So, now actually the teleconference is meant for MSc Zoology students. Your course material on toxicology of insecticides. In this, the topic we have chosen today is botanical insecticides and synthetic insecticides also, that is phosphorus and organochlorine. Now, let us talk about that first. Okay, when we are talking about this toxicology, the very first question is what toxicology is, isn't it? Now, what is toxicology? The father of toxicology itself, he has called toxicology as, it's nothing but, see, all the things which we see are poisonous. Okay, all the things which are poisonous, that means there is nothing without a poisonous effect. That means it is not exactly the thing, but it is the dose which matters a lot. Depending upon the dose, even the things also can act as poison. That means the study of toxicology, we can describe the study as it is adverse effects of both chemical and physical of these agents in the biological system. That we call it as, we can define the toxicology in that way. When we talk about toxicology, we can say that it has both ways, like it is a science and it is also an art. The very first thing, now if you are saying what exactly the scientific aspect of toxicology, it is a kind of it is the first phase. The science of toxicology is gathering the data. that we call it as a science. Okay, then when we, that's the first phase. Now, when we talk about the art of toxicology is like we have the information. Now, the information which we have gathered, we need to predict the likelihood of the adverse effects on this particular dosage, whatever it is, the information which are going to affect on the human beings or the wild animals. This is what we call it as a toxicology. Now, today's topic we have chosen is botanical insecticides and organophosphorus and organochlorines. That means, we are going to talk about botanical insecticides and also synthetic insecticides. There again, anyways, Dr. M. Rajeshri is going to talk about the classification of these things. Now, botanical insecticides means, anyways, as it sounds botanical, it is very safe, because it is a very cost effective and environmental safe, isn't it, Dr. Rajeshri?
Yes,
and same as this botanical insecticides, if you see, they immediately they degrade in the soil,
Yes.
and they will not stay in the
Their persistence is less.
persistence is less, whereas in plant and animal tissue also their persistence is less. As when compare this botanical insecticides with that of the organophosphorus and synthetic insecticides, that's very differs, I think.
Yes.
Now, let us ask Dr. M. Rajeshri to talk about the classification of these botanical insecticides. Dr. Rajeshri,
Thank
can
you,
you give
ma'am.
a
Thank you.
classification, please?
Broadly, these insecticides are classified into two groups: organic insecticides and inorganic insecticides. Organic insecticides are further classified into two groups, that is botanical insecticides, that is what we are going to talk now, and the second one is synthetic insecticides. Inorganic insecticides are arsenicales, fluorines, sulfur. Of course, we are not going to deal with that, but I am going to deal only with the botanical insecticides. The botanical insecticides play an important role in the control of insect pests. The present topic provides the information on chemical substances that which are present in plant material, their mode of action on insects as well as non-target organisms like mammals too. Botanical insecticides are further divided into umpteen number, but we are going to take only 4 of them as a best example. Number 1 is nicotine. I think you can see it on your monitor. Number 1, nicotine. Number 2, pyrethrins. Number 3, neem. And number 4, rotenone. Nicotine is the plant extract of Nicotiana tabacum, and pyrethrins are extracts of Chrysanthemum cinearifolium, and the neem is extracted from Azadirachta indica, and this rotenone is extract of roots of Deris elliptica. Coming to nicotine, nicotine is extracted from tobacco leaves, that is Nicotiana tabacum. It is an alkaloid which is colorless, hygroscopic, pale yellow oily liquid with acrid burning taste. And coming to the physical and chemical properties of nicotine, nicotine is L1 methyl 2,3 pyridyl pyrrolidine, and it is extracted from Nicotiana tabacum. And then second type is non-nicotine, which is derived from Nicotin sylvestris, and anabasine is the third type, that which is derived from Nicotiana glauca. If you take the comparison of the efficacy of these 3 groups, anabasine has got some teratogenic effects, but compared to the other groups, it is teratogenic means they affect at the embryological stages and they cause deformities at the embryonic stages, and larger amount of this is reported to be teratogenic in swines, that is pigs. The chemical formula for nicotine is C10H14N2. This is, you can see the structure of nicotine on your monitors. And coming to the mode of action of nicotine, nicotine is considered to be an agonist. Agonist is that which mimics the other molecule. Say for example, this nicotine has a similar structure as that of ACH, that is acetylcholine, hence they mimic the acetylcholine in both mammals as well as in insects. That is how they activate the some of the most important reactions of ACH, that is neuromuscular activities, cholinergic reactions, and they activate peripheral, central, and also the sympathetic nervous system, both in mammals as well as in insects. In man and animals, it induces both behavioral changes as well as depression. One more important thing is this nicotine, it activates the catecholamines of adrenal medulla, and this adrenal medulla in fact increases the heart function. You can see the side effects of this nicotine on your monitor. Say for example, if nicotine affects the central nervous system, they cause headache, sleep disturbances, abnormal dreams, irritability, etc. Similarly, if they affect the heart, they cause tachycardia, arrhythmias, and coronary artery constrictions. And they also affect other parts of the body, like for example, they affect the endocrine system, especially the insulin producing structure, that is pancreas, and they also affect the blood system, that is increased clotting tendency, lungs causing bronchospasm, they affect muscle causing tremors, and they also affect gastrointestinal tract leading to nausea, dry mouth, vomitings, and sometimes the joint pains too. Similarly, the nicotine has got other effects on insects too. So, for example, in insects like mammals, they also show they mimic the ACH and they interact with the nicotine acetylcholine receptors and activates the ACH. Poisoning by nicotine follow the general sequence of excitation, convulsions, paralysis, and death. Nicotine has a profound effect on functions of heart in cockroaches and even in Spodoptera larvae. This is the general structure of ganglions that which is seen in insects, and this nicotine they affect the ganglionic structures of brain and other ganglions also, so that they mimic ACH and activates the ACH group. Not only this, the nicotine affects soft bodied insects like thrips and bees. Target organism. If mammals are exposed to nicotine through oral, subcutaneous, intravenous, it exhibits high toxic effects. Nicotine is metabolized in the liver into major products, the cotinine, which is generally excreted through fecal matter. Nicotine, if it is found as sulfate molecules, that is nicotine sulfate, it acts as stomach poison for leaf eating insects. Therapy for nicotine poisoning. Nicotine poisoning can be treated by artificial respiration. If it is swallowed, then we can give gastric salvage using slur of activated charcoal or use anticonvulsants, small intravenous doses of barbiturates and diazepam. But one thing everybody should remember, that there is no antidote for a nicotine poisoning, very important point to note down. Coming to the second group of botanical insecticides, that is pyrethrin. And this
1 second, Dr. Rajshri, now you are talking about nicotine.
Yes.
Can you talk about the half-life period of nicotine here?
Yes, sure ma'am. The half-life period of nicotine is only 2 to 6 hours.
Okay.
That is how you do not find the residual activities are not found in the either in mammals or in insects. Coming to the second group of botanical insecticides, pyrethrin. Pyrethrin is one of the oldest organic insecticide, and it is extracted from dried flower buds of Chrysanthemum cinearifolium, and pyrethrins are esters formed by two acids, number 1 chrysanthemic acid and number 2 is pyrethric acid, and this pyrethric acid is seen with 3 alcohol groups, namely pyrethroline, cineroline and jasmolene. Coming to the structure of pyrethrin, you can see it on your monitor. The structure of pyrethrin 1 and pyrethrin 2, it differs by the presence of CH3 group in R1 position in pyrethrin 1 and in R2 position CH2. Similarly, if you take pyrethrin 2, it has got COO CH3 and CH2, and cynarin 1 CH3 CH3 and cynarin 2 COO CH3 and CH3. Target organism. Pyrethrins are used for both insects as well as non-target organism. If it is affected to insects, they act as fast contact poisons. That is, they are the fastest acting contact poisons, and generally they are used as fumigants, but they are never given as stomach poisons. And the most important point over here is this pyrethrin, they directly affect the nerve cell. That is why they cause a rapid knockdown action in insects, very important point. When you compare this point with other group of insecticide, this is called as rapid knockdown acting insecticide, because this pyrethrins, they are synergists. Synergist is when a insecticide is combined with other type of molecule, the metabolic activity of this pyrethrin is activated more than the normal rate. Say for example, the pyrethrin, when it is mixed with vegetable oil, the metabolic product that which is formed from pyrethrin is more and it gets easily metabolized. When you compare the effect of this pyrethrin to mammals, they are less toxic to mammals, because mammals, they can metabolize pyrethrin at a very faster rate and very easily. And one more thing, there is no evidence of carcinogenicity or there is no evidence of mutagenicity by pyrethrins. Even if man is exposed to pyrethrins, they cause few reactions like dermatitis, they cause rhinitis or sometimes allergic reactions, or if it is if the case is more, then it may lead to respiratory failure. Generally, these pyrethrins, they affect on different types of insects. You can see them on your monitor. Number 1, they can affect the mosquitoes, they can affect the cockroach, it can affect your bookworms like Lepisma, and they can affect the ants, etc. The metabolism of pyrethrin. Pyrethrins are easily detoxified by converting them into acid and alcohols by hydrolytic splitting of the esters. This is seen both in insects as well as in man, and they are known to inhibit oxidative metabolism and the pathways involved in oxidation by mixed function oxygenases, that is MFO. Similarly, we got the third important type of botanical insecticide, that is neem. All of us, we know that neem is called as margosa tree, and it has been originated from Burma and India too. And generally, the neem seeds and neem leaves are used for the insecticidal activity, and extracts of seeds and leaves of Azadirachta indica have insecticidal properties. Neem oil from seeds is used for many purposes. Say for example, they are used in soap industry and other industries. We got 4 different components of Azadirachta, that is neem. Number 1 is azadirachtin, number 2 is melantriol, and number 3 is selenin, number 4 is nimbin and nimbidin. If you compare the activities of these 4 groups, azadirachtin is mainly useful in regulating the growth and reproduction of insects, whereas melantriol, it is a mild feeding inhibitor, and the third group, that is selenin, it is a powerful inhibitor. Nimbin and nimbidin, they are used as antiviral agents. Coming to this is you can see the structure of azadirachtin on your monitor. Then target organism. Target organism in the sense, basically this neem has got severe effects on insects when compared to higher animals like man. In insects, they act as ecdysone blockers. Ecdysone blockers are those which inhibit the molting process. Molting is a phenomena that which you see during metamorphic stages. Okay, that is conversion from one stage to another stage is done by molting, and this process is totally inhibited by neem. And they also inhibit fecundity rate. Fecundity rate is the reduction in reproduction rate. And the neem also reduces the lifespan of adults. As already we have seen that neem is a anti-feedant agent. Coming to the toxic effects of this neem to mammals, they are non-toxic to warm blooded animals including man, and there is no evidence of mutagenicity. But one thing everybody should remember, that is, when seeds of neem are infected with a fungus that which is called as Aspergillus flavus, then it has got severe toxogenic effect. Very important point to note down, that is, when they are contaminated with Aspergillus flavus, it has got toxicological effects. Coming to the fourth group of insecticide, that is rotenone. Rotenone is the group of compound that are extracted from roots of Derris elliptica, and this Derris elliptica, it belongs to Leguminosae family. It controls different types of animals. Number one is they act on fish, they can act on beetles, they can affect aphids, they can affect parasites like fleas and lice, that you can see it on your monitor here. They affect on the fish, they can affect on man also, and they are affecting the parasites, and these are the other two groups of insects. And this is the main organism that which we commonly see in our household, that is in books, that is bookworm like Lepisma. The chemical composition of rotenone. The first group of rotenone group is rotoxin, second one is rotenone, dugilin and elliptone. Target organisms. Rotenones are toxic to fish as well as insects. They directly influence the respiration, and poisoning of rotenone is very less in higher animals like man, or you can say very rare also. And if it is given to man or if it is poisoned to man, then it causes dermatitis and rhinitis. Sometimes it can also affect GI, that is gastrointestinal tracts, causing nausea and vomiting. Severe effects of this leads to respiratory failure and sometimes death in man. Effects of this rotenone on insects. Rotenone is used both as a contact poison as well as stomach poison to insects. Poisoning causes inactivity, knockdown, paralysis and slow death, and death is also due to respiratory failure. One important feature I want to show you here is, when you compare with other type of insecticides, the effect of rotenone is very slow. In the previous topic, I was telling you that the pyrethrins are fast affecting chemicals, but here the rotenone, though they cause knockdown, it is a slow process.
I think Dr. Rajesh, you have been talking about botanical insecticides so far.
Yes,
and all the insecticides you have mentioned, do you
Yes.
think they have any effect on mutagenicity or carcinogenicity?
That was that's what I was telling you, ma'am.
Okay.
There was no evidence of mutagenicity or
Okay.
there is no evidence of carcinogenicity
Okay.
till now.
I think so far our students have learned much about botanical insecticides. Now let us talk about synthetic insecticides, organophosphorus and organochlorines. Dr. Ramadevi, can you talk about these insecticides please, synthetic insecticides?
Yeah. Synthetic insecticides are several insecticides are there currently in use, but right now I will be talking on organochlorine and organophosphorus insecticides. And organochlorine insecticides are a group of chlorinated hydrocarbons, and it comprises of DDT and its analogues. lindane and its isomers, and cyclodienes. First, before going into the in detail about these pesticides, let us see what are the general properties of these pesticides. They are lipophilic and highly stable. They contain carbon, chlorine, hydrogen, as well as and including oxygen atoms, and it contains carbon and chlorine bonds, several bonds, and they are having cyclic rings, and which include benzene rings also. They are do not have highly reactive intramolecular sites. So, these are the general characteristics of the chlorinated insecticides, and now we will move to DDT and its analogues. When you look at the DDT and its analogues, DDT analogues are DDD, DDE.
I think Dr. Ramadevi, please show the slide to the students. Yeah, this is the topic which we are going to talk about that. Yeah.
Yeah. This DDT, DDE, dicophol, and DMC.
Yes.
And lindane isomers are, lindane has 7 isomers, of which lindane G is more important, because it has insecticidal property compared to the other hexachlorocyclohexane insecticides. And then when we come to cyclodienes, cyclodienes have are chloraldene, heptachlor aldrine, dialdrine, isodrine, endrine, etc. DDT, its chemical name is 2,2 bis parachlorophenyl 1,1,1 trichloroethane. It has been synthesized in 1874, and its insecticidal property was discovered in 1939 by Paul Muller, for which he has been awarded Nobel Prize in medicine. DDT, it is a broad spectrum insecticide, because why I am saying broad spectrum insecticide is, apart from the insects of the target, it is even toxic to mammals, fish, as well as birds. And DDT is has a more created a remarkable impact in public health programs by it has been used in World War II for mass insectification, for we using a 10% DDT powder, and it saved about 2.5 million people from typhus epidemic. Worldwide malaria control program was also taken by World Health Organization by using DDT in 1955. When you look at the toxicity of DDT, toxicity to the insects, basically it produces DDT jitters, it is called tremors, and because of the production of these tremors, it produces repeated movements in the body, and the insects falls repeatedly, it falls on the back, and then it results in the prostration, and finally it dies. Whereas in the case of mammals, it shows an hyperexcitability, and it creates tremors and then ataxia, and later on it leads to the convulsion, and finally it dies. Whereas DDT has not shown any carcinogenicity to the man. When you look at the mode of action of a DDT, primarily it acts on the nervous system, and it affects on the nervous communication, like it changes the movement of sodium and potassium ion through the axonal membrane. Because of that, repetitive discharge has been will takes place, and this leads to again tremors, and then it produces all the symptoms as I have already explained above. And there are several theories have been explained on mode of action of DDT, but none of the theories have explained clearly about the mechanism of the DDT. So, the theories which are currently being used for the explanation are blood toxin theory, dehydrochlorination theory, conductophore and toxophore group theory, Butterfield configuration theory, rotatability of the benzene rings, van der Waals forces, and Mullin's hypothesis.
Dr. Ramadevi, I think now we have to give a break for the news. After that, we all will meet again.
Okay, thank you.
There is a break now. So viewers, welcome back to teleconference again. Today we are talking about the botanical insecticides and synthetic insecticides. Now Dr. P. Ramadevi, you are talking about the synthetic insecticide.
Yes.
Can you please continue about the DDT?
Yes, please. Yeah, now I'm focusing on DDT metabolism. DDT metabolism takes place in a different routes like DDT, as it is shown in the figure. DDT it converts into dicophol by hydroxylation and to DDE through dehydrochlorination, and it converts into TDE. It is nothing but it is DDD by reductive dechlorination, and it converts into DDA through oxidation. In insects, DDE is the major metabolite is observed compared to other metabolites. This DDE is facilitated, or it is mediated by an enzyme called dehydrochlorinase enzyme, DDT dehydrochlorinase enzyme. Whereas DD dicophol is mediated by mixed function oxidase enzyme. And in mammals, DDA is the major metabolite that is observed, and it is soluble in aqueous media, and so it is been excreted in the urine, and it is through and feces. So, in mammals, apart from DDA, other metabolites are also produced. These metabolites are not to the major extent, but not only these metabolites, DDT is also present in mammals. As high as 12.2 milligram per liter DDT was observed in human milk. So, this is the highest concentration. This shows that there is a bioaccumulation of chlorinated insecticides. Due to this bioaccumulation, the DDT has been discontinued from usage, apart from its public usage in the public health programs. Then we will move to lindane. Lindane is a G isomer of hexachlorocyclohexane, and the hexachlorocyclohexane has 7 isomers. Out of this, lindane shows insecticidal property. So, lindane has been used as a for controlling mites infesting man, such as Sarcoptes scabi and Chaggars. And lindane is highly volatile than DDT, and it shows similar toxicological property like DDT. And symptoms of poisoning, lindane shows similar symptoms of poisoning like DDT. It is also a neurotoxic agent, and it stimulates the central nervous system. When you look at a mode of action of the lindane, it causes accumulation of acetylcholine in the nerve of the insects by inhibiting sodium, potassium, magnesium better than DDT. The lindane is quicker in action than DDT, and also it has got higher vaporizing capacity than DDT. When you look at the metabolism of lindane,
Here one second, Dr. Ramadevi, you are talking about the toxicity.
Yeah.
Do you think that the environmental factors are also involved in this toxic effects of this?
yeah, in the case of DDT as well as lindane, there is a negative temperature coefficient. That means that with increase in the temperature, the toxicity will be reduced, whereas in DDT it is more compared to the lindane.
Oh, increasing the temperature, toxicity is
Reduced.
Oh, I see.
At
Okay.
lower temperature, it is more toxic.
Interesting.
Yeah.
Okay. How about the metabolism now?
Yeah, when you look at the metabolism of lindane in insects, it metabolizes to chlorinated benzene derivatives, and also glutathione conjugates. These glutathione conjugates, again it hydrolyzes to give thiophenols, chlorinated thiophenols. Similarly, when you look at the metabolism of lindane in mammals, lindane metabolizes and it produces several chlorinated phenols, and this is like mediated through by hydroxylation and dechlorination and conjugation. And then we will move to cyclodine insecticides. The cyclodine insecticides we have already seen the heptachloraldrin, dialdrin, and endosulfan, all these fall under cyclodine insecticides, except endosulfan. Other insecticides are banned because of their high fish toxicity and long persistency. Mode of action of insecticides. These insecticides, like unlike DDT and HCS, as I have just now I have explained to Dr. Neti, here reverse is observed, like as temperature is increased, the toxicity is also enhanced. This is the difference between DDT, HCS and also cyclodiene insecticides. And another differentiating factor is cyclodiene, they are skin, they have express skin toxicity also, unlike other HCS and DDT, which are oral toxins mostly.
Dr. Ramadeva, as an analyst, now you should be having more information and idea about this. How do you think we treat this OP and OC poisoning? How to treat that?
Yeah, this
The
chlorinated,
OP, the organophosphorus, and the OC is
organochlorine poisoning is treated by anticonvulsants like
Barbitrates. Barbitrates.
Phenylbarbital.
Okay, I see. Okay.
One sec.
And diazepam can also.
Phenobarbital and pentobarbital and phenobarbital, and also by diazepam it will be treated. Whereas when you look at the organophosphorus insecticides, it is treated with 2-PAM as well as atropine. Those two will be used for treating the organophosphorus poisoning. That is basically how they do it, is like they treat this excess of acetylcholine that is produced,
ACH.
that is neutralized by using these anticonvulsants like atropine. And also, like another way of doing is by bonding, like it forms a complex with the phosphate, this organophosphorus insecticide. So when this PAM is used, it releases the enzyme from the complex form from the phosphate which forms with the enzyme. So when it liberates phosphate from the complex, then enzyme will be liberated, so that will be detoxifies that way.
Okay. And what are the actually to see the estimation of residual?
Residual.
How do we do the? What are the methods we can use?
Yeah.
Analytical methods I'm talking about.
Like methods like GCMS we can use, and high resolution GCMS for identifying these residues.
What is GCMS?
Like gas chromatography, mass spectrometry.
Because I want my students to understand what exactly.
Yeah.
Okay.
So that will be used, and as well as you can even use LCMS.
Okay.
And GC with electron ECD detector, electron capture detector. So there are different methods are available based on the type of residue we want to analyze in the whatever is the matrix. Like from that matrix, first we have to separate it by extraction and pre-concentration. We have to pre-concentrate because of the lower concentration, the detectability of the instrument also based on the detectability, and then we will be analyzing by different instruments based on the type of residue we want to estimate in that.
Generally we can even estimate from the water and the plant, the stems and etc. Right?
Yeah, it can be. We are doing from food as well as from fruits, vegetables, and water and soft drinks.
Okay.
All that we.
So before they send it to the market, these kind of tests are done now.
In wherever it is manufacturing is done, people do that, like different industries, whichever like soft drink industries or like distillery, drinking water industries, all them there is a norm, like they have to follow those standards if they want to market in the.
That's right. Yeah, nowadays I think now it has become very common in India. Like in markets we are finding the tinned food and everything.
Correct.
And we do not know whether the the declaration is authentic or not. So this kind of tests ought to be done, right?
Correct. Yes.
So what are the what is your contribution in that? Your companies, are they testing these things, products?
In all wherever we do it, it will be tested based on the requirement where we are going to export and where we are going to market. Like each country will be having a different standards.
True.
So based on their standards, we have to meet the requirement, like concentration limits and all that.
Okay.
So that will be done, and then.
That's true.
It will be like we have to issue a certificate to the concerned authorities, like a company wherever we are supplying
market.
or whichever is.
Yeah. Okay, I think now she has explained about synthetic insecticides.
Yes.
Can you talk about that bioaccumulation of insecticides, Dr. Rajashree?
Yes, of course, ma'am. See, when compared to botanical insecticides and synthetic insecticides, botanical insecticides they do not show any biomagnification. As it is till now, there is no evidence of biomagnification or bioaccumulation. Biomagnification is the tendency of the pollutants to concentrate as they move from one trophic level to another trophic level.
Yes.
Okay, but that is not seen in botanical insecticides.
Oh.
But I think in synthetic insecticides,
Yeah, because the
they do
environmental
cause-
factors
Yeah,
also she mentioned,
yeah.
no, with the temperature it varies.
Yes.
So that's
they
true.
do cause a biomagnification.
Like there is one slide I can show that.
You
Yeah.
can see
This
it
is
on
the
the monitor.
slide. Like
The bioaccumulation is again, it is defined as entering of the pollutant into food chain.
Oh,
Food chain
In
and
this-
their increase from the environment from first organism to the in the food chain.
food chain.
Yeah.
Yeah, next organism.
As you just go from lower level of the food chain to higher level of the food chain, so you can see it on your monitor.
magnifying concentration levels by
Of
10 million
DDT.
times. Yeah.
DDT, it shows DDT effects will be around a 0.003 ppm when it reaches to the microorganisms like phytoplanktons,
Yeah.
and when it is passing to the small fish and zooplanktons, it will be around 0.04 ppm, but once it is reaching to the fish 0.5 ppm, but again large fish it will convert to 2 ppm, and the fish eating birds they show around 25 ppm of DDT. That means you can see the accumulation of the chemical, it's going on increasing as the food chain is going to the top level.
I think even the DDT is now totally banned, right? That's the reason why organochlorines have come to the picture now.
Organophosphate is
Phosphorus,
compounds. They have come into the picture.
Oh, okay, yeah. DDT is organochlorine. True.
Organochlorine.
So that's banned totally.
Yeah.
Yes.
No, but they are using only in the public health programs for controlling like insects and some which are not of much where we don't give that much concentration. Like.
Oh, okay.
And most
Yeah.
important thing is you don't see the persistence of the chemical, botanical insecticides.
botanical.
Yeah. But
Yes.
here you find the persistence of the chemical in the environment. That is the basic difference between the botanical insecticides and synthetic insecticides.
Yeah. So Ramadavi, you can talk about
Yeah,
some
organo-
methods to
yeah.
analyze this.
Yeah.
That's more important for us.
Yeah, we can do this analysis by GCMS and HPLC methods, but this GCMS before analysis we have to pre-concentrate that by doing some extractions, like for example if we take in the water that we extract it with hexane or dichloromethane, and then we pre-concentrate it, and that we will analyze it by different techniques like GCMS or HPLC, LCMS. Then we will measure what is the concentration level in that.
What are the
But
methods here?
I think all these equipments it will be seen in a well settled labs.
Yes.
But when compared
Research
to
labs.
the research
That is what
labs,
I'm asking. What are the methods you are
yeah,
using?
but if
in
you
the-
talk at the level of university level, I don't think these instruments are useful.
Yeah,
but
chromatography, of
yes,
course. Yeah.
low cost effective
Yes, that's
instruments can be used. Say for example, column chromatography and flame photometry. All these things I think we can use it in at a university level also, and they are cheaper, and of course they also give the accurate results. And another most important thing, I think electric conductivity will also give the testing of these residues in water. This is one method of analysis of the residues.
Okay. And when you were talking about the botanical insecticides,
Yes.
I think in the rotenin,
Yes,
respiratory effect of it,
Yes, very
I
good
think we
question.
left it,
Very
yeah.
good question.
yeah. Can you talk about that?
Yes, sure. See, this rotenone, it gets converted into or it gets metabolized into two groups. The most important one is rotenolin 1 and another one is rotenolin 2. When compared to the parent molecule, rotenolin 1 and rotenolin 2 are less toxic. Similarly, we got one more metabolite of rotenolin, that is hydroxyrotenone, which is as toxic as that of parent molecule. Coming to the effect of this rotenone, rotenone is a respiratory chain inhibitor. We know what is respiratory chain. Respiratory chain is a sequence of reactions where you find the energy flow is there from substrate molecule to half molecular oxygen, and at this level we get 3 ATP molecules through this respiratory chain, and rotenone has got the severe effects between FMN and CoQ, and rotenone can inhibit the respiratory chain at the level of CoQ and FMN. Similarly, the other types of inhibitors that affect the respiratory chain is antimycin A, that causes inhibition at cytochrome B and cytochrome C1, and finally, the cyanides. The cyanides can affect from oxygen to half molecular structures. Okay. That means, basically this rotenone is used both as insecticide as well as pesticide. That is the most important, and commonly this rotenone is used in some countries in the early days to catch the fish.
Oh.
But they were not knowing the exact action of this rotenone, but to catch the fish they used to use this rotenone as a bait, and once the fish attacks to it, they used to collect it and eat it.
Okay.
That was the history behind the rotenone. Like that they have been used as a pesticide, but
So
later
even in India, we have been using
India
it? Not exactly.
not exactly.
Okay.
And this rotenone, it generally depresses the respiratory functions, and they also change the heartbeat rate, and the primary effects is only the respiratory chain, both as in insects as well as in man. Okay. That is why it is not generally used for as a common insecticide.
I see. Okay.
Basically, it is used only in fish culture to eradicate the unwanted fish, predatory fish. Rotenone is used.
Okay. So even though we know that the botanical insecticides are much better than the synthetic insecticides, anyways, all the synthetic insecticides are not eradicated, right?
No, no,
They are
they are
still,
not.
you know, we are using
Organic.
that.
Still we are using it.
Yeah. So how far you justify that then?
I think everybody should be properly advised or allow the people to know about
Yeah.
each insecticide so that they can
Yes.
understand the role of each type
Exactly.
and classify themselves as whether it is toxic or non-toxic, botanical or
True.
synthetic, and they can classify themselves which is more toxic and which is not toxic.
Yeah. Mostly we even see that, you know, people who are using these, they don't even wear the mask and they don't even use the, you know,
Another,
the gloves.
Yeah,
and
the
I think a
precautions.
lot of awareness we should create among
Yes,
students,
precautions,
right?
yes.
That we should be given
Yes.
for all the people who are doing job with this kind of insecticides. And I think normally we also see that, you know, the DDT, they spray near our houses also.
Yes,
And how dangerous it is then.
they are dangerous.
Yeah.
But common man, he should be properly educated so that they can reduce the usage of
Exposure,
these
yeah.
chemicals and let them protect themselves with suitable.
Yeah, but now we are
agents.
seeing that, you know, every week many times they come and spray in and around our houses. So immediately we cannot wear our masks and sit. And what is the concentration they are using actually now? Have an idea? Is it really very dangerous for?
No, no, no. It will
not,
be very low concentration.
Very
like,
low concentration they
yeah.
use. Yeah. Nowadays I think we have lots of mosquitoes anyways. For that, you know, mosquito spray they are using. Yes, and any more you want to talk about these insecticides
Yeah.
and the important questions, or you want to give advices to our students? Please go ahead.
Yeah,
Ramadhan.
these organophosphorus insecticides I have not completed actually. But they are the major advantage with organophosphorus insecticides are they are biodegradable. These
Oh,
insecticides, the
yes.
major use of these insecticides because of the bioaccumulation of the organochlorine insecticides. These insecticides have come into the picture. They have been synthesized, these insecticides, and they are very acute toxins, and they are not biodegradable, and so they are safe to use compared to the organochlorine insecticides. And these insecticides, like complex with acetylcholine, and because of the acetylcholine complex, they will exit their toxicity levels.
One thing common what I have seen is, ma'am, like let it be synthetic insecticide or botanical insecticide,
yes.
the main target is on ACH.
And that is true.
Yes,
So
ACH.
everything is on ACH.
ACH. That means directly they have got some impact on nervous system.
Nervous system, true.
Because especially in insects, in order to make the insect immobile, I think first the target should be on ACH. The same thing is seen both with botanical insecticides as well as with synthetic insecticides.
Yes, okay.
That is the most important feature what I feel over here.
Yeah.
So what are the general important questions students can expect?
The most important questions for PG students, I believe, the classification of insecticides, number one, and number two is the methods how to analyze the residues in let it be in water or any other component about this insecticides, and third question is how to control the OP poisoning, or the OP poisoning can be treated and how this is done. These are the most important questions, I think, and everybody should know it.
Yes.
Because not only for the sake of exam, everybody should know how
True.
these OP components can be treated.
Yeah. So Dr. Rajeshri and Dr. Ramadevi, thank you very much for participating in today's teleconference. I think my students have really got the advantage out of it. That's the reason why we invited different people on this dais for all my students to interact with you. If you still have any doubts, please come and contact us in the university campus. Okay, bye bye.
Thank you.
Thanks.